Hi students, so let's continue with mutations and now we go into the next session called the frameshift mutations. As a result of certain point mutations, what happens sometimes the reading frame of a gene will be changed. Now you know that a DNA will code for an mRNA, and mRNA is being read as triplet codons, as 3 unit bases, as triplet codons. They have been read by the what? By the ribosome, and they synthesize proteins. Sometimes what happens when you add some DNA onto a sequence, or when you lose some DNA sequences from the DNA, what happens? This reading frame of the DNA, that is the triplet codon reading frame of the gene, will happen to change, and that will result in the shift of the grouping of these bases, and the change in the amino acid also will happen. And such shifts would result, that shifting mutations are referred to as a frame shift mutations. And as a result, what happens? The resulting protein will be usually non-functional, and different things can contribute to frame shift mutations. It can be either insertions, it can be other deletions, and it can be duplications. Some people also call it as indels, that is, insertion and deletions together contribute to frame shift mutations. But sometimes we can also find that duplications can also result in the formation of frame shift mutations. Let's go into the details. Now, frame shift mutation is also known as framing error, or also a reading frame shift. Different names have been put forward to it, and sometimes we can also see that by shifting the reading frame, what happens? It would also the first stop codon, and even when the full protein has been formed, what happens? What happens, this protein synthesis might get stopped or something. So basically, by adding or deleting some nucleotides into the bases, what happens, the reading frame of the protein synthesis, during the protein synthesis, for the protein synthesis, the reading frame which has been needed, that gets altered, and as a result the protein also gets altered, or a different functional or a malfunctioned protein can result. I guess it's quite not understanding right now, if unless and until you see a figure. Let's see into this with a basic example. I'm not talking about, in this example we are not talking about the proteins, but look at a sentence. Matt got the red hat. Okay, here I'm changing it. M is being changed into Pat, P. So it becomes Pat got the red hat. Okay, then Matt got the red hat. R is being changed to B. So it becomes Matt got the bed hat. Then next one we had, I'm changing H. So in the triplet codon, one you should remember, in the base sequence you have corresponding to that you have a mRNA sequence. In the mRNA sequence, if anything is getting changed, the sense of it itself gets changed. Now Matt got the red, instead of H, if you put S, it becomes a Matt got the red sat. So all these are examples of what? Point mutations. And if you're losing one letter, it becomes, so this is a substitution, okay? And what happens if you're deleting something? Now here, Matt G is gone. So Matt ought her ed at. So a loss of G will result in the shifting of the frame. See, that is, one has been removed from here. So this OTT will come to the side. So a deletion will result in the formation of a frame shift mutation. Then if you're having an insertion, now Matt, instead of after GOT, I'm putting an L here. It becomes Matt glot. It makes no sense. So by adding or putting something, this is an insertion and this is a deletion. By the addition or the deletion of certain base sequences, what happens? The reading frame of the particular peak sequence will get changed. Now this is an example of just a letter, a alphabet, or a sentence. Alphabet is being changed, the sentence becomes meaningless. But in the case of genes, what happens? When you change one nucleotide base, when you add one or when you delete one nucleotide base, the amino acid sequence which it codes for will get changed. Mainly because the reading frame has been changed and the amino acids which it adds will change. And as a result, you get a malfunctioned or a non-functional protein. Let's go with amino acids examples right now. Now here, this is a normal mRNA. and it codes for a protein with methionine, lysine, phenylalanine, glycine, isoleucine, valine, and proline. These are the amino acids which have been coded by the normal mRNA. But if you come to a frame shift mutation which has been caused by a single, now here a deletion of U is been happening. So what happens when, remember, this is the triplet codon, this is the triplet codon. Till here, what happens? It is fine because the codon is the same. Methionine and lysine has been formed. And then when one U is gone, it becomes UUG, and that will code for mucin. And then it becomes, the codon is changing. And GCA, it codes for alanine. So instead of phenylalanine, here leucine comes, and instead of glycine, here alanine comes. And instead of isoleucine here, and a stop codon has been initiated. So that's why this is an example of one deletion of one nucleotide base will shift the reading frame of the mRNA, and as a result, the protein will be changed, and sometimes even beforehand the stop codon can come. It can result in a nonsense mutation also. Now this is another example here. This is a original DNA sequence which where a strain of histidine molecules have been synthesized. Okay, and into this there is an insertion of adenine here in the DNA sequence. So you can see that after 3 histidines, it becomes threonine, then serine, serine, serine. So incorrect amino acid sequence are added and the function of the protein is being changed here. So by an insertion or a deletion, what happens? Sometimes the frame shift or the reading frame of the mRNA sequence will be changed, and as a result a different amino acid was added, and a different amino acid could result in the formation of a non-functional protein. Such mutations in which the reading frame of an amino acid sequence or the amino acid or the reading sequence of the mRNA sequence of the mRNA which codes for the amino acid has been changed. In such cases, such mutations are referred to as a frame shift mutations. Now, what has this got to do? We might think that this is not going to affect us, but just have a look of some of the mutations and what are the diseases which have been associated with it. Some common examples which we have now. Just look. If you have a mutation in the case of breast cancer, you have two genes which are mainly responsible for the incidence of breast cancer, and BRCA1 and BRCA2 are some genes. Now BRCA1 is a gene on the chromosome 17, and you can see that in the chromosome 17, the BRCA1 gene, when it undergoes nonsense, frame shift, missense mutations, what happens? Okay, see, just these colors indicates the sites for nonsense and frame shift mutations, and the yellow color, it indicates the sites for missense mutations. So as a result, you can see that in the BRCA1 gene itself, various mutations have been reported, and some of these mutations, okay, so you can see a nonsense in a frame shift mutation which is resulting into a nonsense, that is a stop codon, and that itself will result in the incidence of breast cancer in those people. Those people have a greater incidence of breast cancer. And here BRCA2, which is located on chromosome 13. Okay, here it is having all the green colors are the nonsense of the frame shift mutations. At different locations of the chromosome 13 itself, they have lots of nonsense and frame shift mutations, and also they have missense mutations. You know what's a missense mutation? A missense mutation is which and one amino acid is replaced with another amino acid and the protein becomes non-functional. So these are the indications where mutations can result in the formation of various diseases. Now, you might have a question like, is it an exclusive example of a frame shift mutation? Isn't an example of a nonsense mutation? Or is it an example of a missense mutation? Most often it has been found that various diseases are being linked to it might be due to the cumulative effect of various mutations. Now, coming to HIV, which we have here, HIV, in the case of HIV infection, it has been found that a 32 base pair deletion in the CCR5, okay, it's a gene, okay, it always negates to the likelihood of an HIV infection. Okay, and next one we have is another disease called the Smith-Magenis syndrome. Okay, SMS. Just remember Smith-Magenis syndrome. Okay, this is a you have intellectual disabilities, sleep disturbances, behavioral problems, and a variety of craniofacial, skeletal, and visceral anomalies. And it has been found that about approximately a 3.5 megabillion of deletion has been found in a particular gene here, and that is resulting in the particular disease called the Smith-Magenis syndrome. Okay. And also, I told you frame shift mutations could also be caused because of the duplications. Now, for example, you have, and they are also called as a repeat expansion nucleotide repeats. This is another term which has been given for the a type of duplication. And here, it's one example that we have is CAG repeats. The CAG repeats is responsible for about 10 diseases like Huntington's disease, spinal and bulbar muscular atrophy, dentatorubular pallidoluysian atrophy, and seven other diseases. Okay, and when the sequence of about CGG is being replaced, what happens? It will result in the formation of a fragile X chromosome. So these are all mutations which will contribute towards either at the protein level it will happen, protein changes can happen, or even you can see in this the X chromosome itself is getting affected. Okay. So these are about the frame shift mutations. I hope it is clear to you. Thank you.